you, Evan. Great to see everyone this morning. Sorry, I can't see your attractive faces on Zoom, but I'm glad to have all of you here to talk about the sexiest topic, monitoring. I know you have all been waiting with bated breath throughout this entire workshop to talk about the coolest thing, the most well-funded activity, monitoring. We have assembled a fantastic suite of speakers for you today. We've been talking, all day yesterday and all day today about the functions and the values of marsh migration space, why transition zones matter, their history, their present, their future. It is, for all the jokes, it is important to recognize that we know what we know about the value of transition zones because of monitoring. And particularly if we're looking to support those transition zone functions now and into the future, if we want to create that space for wetland upland connectivity, for wetland floodplain connectivity, we have to understand how those systems are functioning, how they are changing, how they are responding to environmental drivers. And we have to do that in perpetuity, right? I really appreciated Peter yesterday. Well, you guys know I always appreciate when Peter speaks, but I really specifically appreciated his emphasis on observation, on not just looking but seeing. And I think particularly when so many of us are stuck behind screens and relying on remote sensing and checking out things in Google Earth and ArcGIS all the time, that we can forget the value of really getting our boots and our chacos and our tivas muddy and seeing, not just looking, but seeing. And so that's what we're going to talk about today. Got some really fabulous speakers. So excited for all of them. So we're going to start with Julian Wood and Josh Nuzzo from Point Blue Conservation Science. Julian is the San Francisco Bay Program Leader at Point Blue. Josh is the Nursery Manager. And do the two of you want to take it away? All right.
So thanks everyone for coming. Thanks to all of you online. I wish you were here, wherever you are, in person. But yeah, so we're gonna co-present. I'm gonna co-present with Josh Nuzzo, and let's get my slides up. There it is. Great. So we're gonna be talking about communities restoring the wetland edge, and just a quick little menu for the day. I'll start you out with some pickleweed salad here. Josh is going to tell you about the story of straw. And then that's going to lead into their work in the transition zone and him asking me the question. And then I will spend a few slides attempting to answer that question. We'll be talking about the T-zone monitoring methods, very briefly, sorry. And some of the results from that study and what some of the recommendations were that came out of that study. And then turn it back over to Josh to look forward to how we've taken those results, those monitoring programs, and essentially that project and kept it moving forward, integrating with the straw team. So he'll talk about that restoration and study design, and also how they're integrating community science into that work. So with that, we turn over to Josh.
Okay, here we go. I'm nervous. Well, I just like to rewind a little bit about 30 years ago to just kind of like tee us up to why I'm here presenting to all of you today. And it all really began in a 4th grade classroom in Marin County with our founder and ambassador, Lorette Rogers, then a teacher, asking the question of what can we do to respond to endangered species? The students quickly became very distraught and very depressed. And one thing that the students did do that I often don't is they were acting locally and thinking globally. So when they want to address the issue of endangered species, what they did was they adopted this beautiful beast here, the California freshwater shrimp. And after some research and some advocacy, they found that the California freshwater shrimp, which is endemic to the creeks and streams of Marin, Sonoma, and I think Napa, County, that the reason why they were endangered is because of habitat loss and land use changes. Yeah, so from there, they planted willow sprigs and worked with private landowners and ranches across Marin and Sonoma. And since then, the project has really evolved from the humble shrimp club of fourth graders to who employs me now, which is STRAW, which stands for Students and Teachers Restoring a Watershed. And although we've greatly expanded and 30 years has passed, we still hold those initial values, which I hold in my heart, but also on this notepad. And those values, although our scale has changed, look at these awesome numbers, we still work to empower communities to heal the land, revitalize habitats, generate cleaner water, sequester carbon, empower children, train teachers, and inspire conservation leaders. And since that one depressing day in that fourth grade classroom in 1994, and now this is out of date because restoration season has started, but we've engaged with over 60,000 students, 10,000 volunteers, largely adult chaperones, conducted over 750 restorations, installed 40,000 native plants, and restored over 12 acres of marsh upland transition zone habitat. So although our pace and scale have largely changed, we're still working in riparian areas in Marin and Sonoma, and now San Benito and San Mateo and Alameda and Plumas County. We've also expanded to working along the bay's edge in all those counties. So we know that if you install native plants on the landscape, you have that like phrase that like, if you build it, they will come. And that's so true. That's so true. But since then, Straw has merged with Point Blue. Wow. And amazing. And conservation science is like at our core. So when we were working with Point Blue, we know that installing plants, especially rhizomatous ones, on the levees or along the bay's edge, we knew that was a good thing, but we were wondering how can we directly address the issue of the ridgeways rail, and how can we like refine our restoration to better like bolster their population numbers and better accommodate them in our changing landscape. So we turned to Julian and we were like, how do we do that? And then Julian said,
let me get back to you on that. Right. No, it was really kind of a simple question. They just wanted to know, well, we're restoring this T-zone, but we want to know like what's the impact on the marsh birds that are over there, T-zones over here, and how can we show that like in proposals or when we talk to people, like what's the one-to-one benefit? It's like, oh, yeah, ridgeways rail. They need the T-zones, upland high tide refugia. They go in there to hide when the water comes up, and it's good. Cool. Are there any papers we can cite, or like how beneficial is it? And then like what makes it good? And so that's where I kind of broke down. I did a little research. I didn't really find anything. And so that's when I decided we needed to address this question with a study. And so I'm going to spend a few slides talking about that. So some of those questions were, I guess the ultimate one was like, how do ridgeways rails benefit from transition zones? But some of the other questions were like, what features are beneficial in the transition zone? What is it about those areas that are most beneficial? And what other birds are in the transition zones? We didn't really know that either. Like there just hasn't been a lot of work in this like narrow little habitat. And then finally, like what are the restoration recommendations that can come out of that study? So this isn't just an intellectual exercise. We've got people doing work on the ground. How can they do it better or keep doing the good things that they're doing? So those are some of the questions. So we started this project affectionately called T-Birds, and worked with Straw to set up this project. This was with funding from the National Fish and Wildlife Foundation, and then we also worked with Save the Bay and San Francisco Bay Bird Observatory too. So they helped us kind of set up the study design and develop the protocols that we used for this particular project. So these were our study sites. I'm not going to spend a lot of time here, but it was a mix of levee restoration, transition zone sites and natural transition zone sites. Some were restored, some were not restored, and it kind of all over the bay there. This is a schematic of like a typical site. So you have the transition zone and marsh in front of that. And then these yellow dots represent our point count surveys that are in the marsh for the birds that are breeding in the marsh. Ridgways, rails, song sparrows, common yellowthroats. Those are 5-minute point counts, getting a little monitoring details in there. And then we have the transition zone, and we divided that transition zone up into blocks, into 200-meter blocks. And within that, we, well, if I go to the next slide, yeah, have the protocols. We conducted vegetation surveys and also bird surveys within those blocks. So these are the main protocols I'm going to talk about that we developed: the winter vegetation assessment and the marsh bird surveys. So that's both the secretive marsh bird surveys that happen kind of in the winter into early spring, and then also the breeding, what I'll call breeding marsh bird songbird surveys. And that happens a little bit later in the season. And those surveys have been going since like 96-ish, 96 to 2005 is kind of when those were developed. And then we had the transition zone bird survey within those plots in the transition zone. So that's we used a simple area search survey. So that's like a 20-minute walking transect type survey. Very easy to implement. And then we had a spring vegetation survey. Let's see, what else can I say about that? One thing that we wanted to do with these protocols is keep them relatively easy, so that even like non-professional scientists could implement them, as long as they knew their plants, which the straw team does. These could be implemented. And I won't go into too much detail on those protocols, but let's see. I think I already talked about the bird area search 20-minute survey. We get the number of individuals by species, record breeding observations, predators. So again, that's within that transition zone. Spring veg surveys, we did photo points, collected percent cover of different cover types, percent cover of dense veg. I'll pause here. So dense veg, like what the heck is that? It sounds vague. We hypothesized that rails and other marsh birds needed dense vegetation to hide during these high tide events when they're going into the transition zone. So we just defined dense veg as difficult to see through and within the 30 centimeters of the ground. And so if you stuck your graduated meter stick in there, if you couldn't see the notches below 30 centimeters, that's dense vegetation. In a nutshell. There's more details in the protocols. The link is at the bottom of the slide there if you want to see that. And definitely let us know if you're interested in doing any of these, because I can offer some insights. Width and slope, percent cover by species, and plant height were other things that we measured as part of the spring vegetation surveys. So these were some of the common birds that we found in the transition zone. So on those area search surveys. Beautiful birds. And then of course, we also detected marsh birds in the transition zones as well. We all know, you've probably seen song sparrows perched on baccaris, on the edge in the transition zone. So those are some of the common birds there. I did not include the ridgways rail here because it's not necessarily a common bird that you see in the transition zone. They do use it. I have seen them there, but just not particularly common. Okay, now let's get into some results. So this one is looking at the transition zone area search bird data set and looking at the impact of dense vegetation. So again, that's dense vegetation that's within 30 centimeters of the ground. So the total number of individuals that we detect is positively correlated with dense veg. So more cover of dense vegetation in your transition zone, you get more birds, basically. And you can, so along the X is the percent cover of dense veg at that site. Okay, sorry, I'm kind of going through these pretty quickly, so apologies, but try to simplify here. So this one, we looked at song sparrows. And so just to reiterate, this is looking at the data from the tidal marsh, relating data from the tidal marsh birds to the transition zone characteristics. So kind of two like separate areas, which I honestly didn't really expect to find much correlation between those two, but we did. And song sparrows were also positively correlated with dense vegetation over 30. So on the, just to walk you through this on the Y, this is population trend. So it's kind of our indicator of like population health. Above 0, the population at that particular site is growing, it's increasing. Below 0, it's declining. So over in all of these dots represent the different sites, values for different sites. So we can see there's a pretty sharp increase in like population health, increasing trends. When you get dense vegetation, that is, at least 10, 15, ideally 20% cover of the transition zone site. So we also looked at the ridgeways rail, and here we found that maximum vegetation height within the transition zone was important for them. So again, ridgeways rail data from the marsh compared to characteristics in the transition zone. And here we're also seeing kind of a increase in benefits, like right around 40 or 50 centimeter tall plants. So, something like that. And then the benefits kind of start to decrease a little bit after that. So that's for the ridgeways rail. And then ridgeways rail also positively correlated with transition zone width. So this one was kind of cool. Basically showing like around a little bit, I mean, the benefit basically keeps increasing with transition zone width. So the take home message is really like, you can't build it, too wide. Obviously, we don't always have that luxury here in the Bay, and despite, so I also want to reiterate, like even short, steep, levee transition zones, we did find to be beneficial. So it's still worth it to invest in that type of a transition zone, but wider is better. So some of the recommendations that came out of this work. Restore for dense vegetation greater than 30 centimeters, and try to have at least 30% of that transition zone area covered in dense vegetation. Plant taller plants about 50 to 100 centimeters in height, so like about waist high. Higher than that, they start to become predator perches. So we want to discourage planting trees and really tall veg in the transition zone. Design for wider transition zones. And don't focus on a single plant species. I didn't present these results, but we looked at specific plant species and there was no silver bullet, like plant this and it's great. It's really more about the structural diversity, the height, than the density. And then finally, we got some mixed results on grasses, indicating that they might be beneficial. It was hard to tease apart from transition zone width. Those two variables were highly correlated. The more natural, wider transition zones were dominated by grasses. So now we're saying, don't remove grasses. They might be beneficial. And we already know they're beneficial for things like salt marsh harvest mouse. So even for that reason alone, it might be a good idea to not remove that. So we had some questions at the end of this study, and specifically about that grass. Like we kind of wanted to dig into that. How beneficial, is it really beneficial? What types of grass is beneficial? How much? Should it be interspersed with shrubs? Bottom line, we just wanted to know more about how we can improve the restoration on the ground. How can we restore these transition zones more efficiently? Kind of goes back to the grass. If grass cover is really beneficial, that's way easier to do than these intense, dense shrub plantings. And the straw group can cover much more ground if they're planting native grasses. And then also we wanted to look at just native plants versus invasives, just generally. That's a question for straw, since their focus is obviously on native plants. Okay, so I basically threw the questions back at Josh. So he's going to talk a little bit about taking those questions going forward and how we're continuing to implement these protocols on the ground and designing restoration sites to answer those questions.
Yes. Isn't that nice when you ask a question and you get a question as a response? That's a cool behavior. Cool. So a lot of these findings from this initial study were like, yes, good, great. Big transition zones, good. Grasses, good. So what we were doing is so beautiful, Hamilton, right? So what we did was, I think in like 2017 or 2018, we were granted Measure AA funding. Thank you, funders, if you're here. And my note. And so the Measure AA funding was to help us conduct and enhance restoration around the SF Bay, to restore both tidal marsh and upland transition zone habitats. And we're like, wow, that's what we do. Amazing. So what we have here on this really legible map is we have these really great sites that are ready for restoration and ready for us to implement some sort of study design to further our understanding and all the nuance of Julian's recommendations. So we have a few here, and they're really easy to see. They're indicated by these beautiful binoculars. One is Schollenberger Park. So cool, go there. That's where our office is. Please go. Another one is at the San Pablo Bay National Wildlife Refuge, the Dixon unit. We heard about it a bit yesterday, how the levee was falling into the bay. Bummer. And then we also had one at the Bahia Wetlands in East Novato, partnered with Marin Audubon. So at these sites, they're levees. And they were kind of denuded of vegetation or filled with invasives and non-native species. So what we wanted to do, being straw, right, we want to work with students and teachers, and we want to restore that watershed. So, and with Julian's recommendations, we're like, okay, so what do we do with all this information? And so we generated, in conjunction with our partners, this study design. And it is really wild looking, and it is as wild as you think it is. So what we did was we understood that dense veg was important, diversity in structure and diversity of species was important, but then we're like, but how do we get the students to plant these in the way that we like? So what we did was we pulled up an old Excel and created this beautiful Tetris. So right here, we're seeing each cell is about 1 meter by 1 meter. And we calculated that we could probably do across these sites about 50 meters at a time with this intensive planting. And each color is a different species, and each cell is a different individual plant. So what we did was a paint by numbers with the students out on the levees, and we laid the flags out just like this. And it was super easy and really fun, and you should do it too. And so we have right here, we have our study design, which is the diverse shrub forb plot with some grasses. And then we also have a grass only plot, which is so refreshing to lay the flags out for that. And you can see, we could talk about all the different species, but these are our favorite competitive weedy rhizomatous native species that we love. And then our grass plot is, our Elemus triticorides and Gesticula spicata. And so we implemented these study design across all 3 restoration sites and got students to install the plants. Being straw, we had about, I think over the course since 2018 to just last year, we had about 45 unique schools come out. Each school had about 2 to 6 classes, and each class has tons and tons of students. So it was a really big effort across a long period of time, or long for me. And so we had those installed. And then of course, we're continuing Julian's rigorous, scientifically based monitoring. And being straw, we wanted to science and being straw, we wanted to get community involved in all aspects of the monitoring So through the summer, we have this community college conservation internship called CCCI, where we get community college students, primarily populations that have been like left out of conservation, to work with us for 6 to 8 weeks. And so we had them involved in the spring veg monitoring and other things like that. We also revamped our internship project, and now they're apprentices, and they help with the monitoring as well, doing spring veg, dense veg, which is the funnest one, and area search surveys, to continue to build on this data set to help our understanding of like what is actually helping the ridge-waste rail populations. And there's us having fun. And so we're continuing to do these monitorings, and we have some take-home messages, and they are as follows. Healthy T-zones benefit tidal marsh birds. Whoa. Yeah, done. Restoration using study design improves our understanding, and community-based restoration has lasting benefits on the landscape, but then also within the human communities that surround the Bay Area. I think that's it. Thank you.
That was fantastic. Thank you, gentlemen. I cannot wait until my daughter is old enough to participate in straw. She's going to be insufferable. Can you even imagine?
When do we do the science? You're doing it now.
All right, so I think we're going to quickly switch slide formats, and we're going to bring up Katie Smith from WRA. Katie is, I want to make sure I get her title right. She's a senior wildlife biologist, but most importantly, she also refers to herself as lead weird mouse girl. I really think that we should all begin to refer to ourselves, like have an alternate title. So thank you. I learned that she has captured 5,000 salties in her career so far, salt marsh harvest mouse. I did not realize there were 5,000 salt marsh harvest mouse to catch in the estuary. Are you sure you haven't caught the same one, the same 1,000 times?
Probably not.
Okay.
I think there's actually about 1 million salties.
Wow, that's amazing. That's a lot of endangered species. Okay, so she's going to talk to us about salt marsh harvest mouse. Take it
Thanks.
away.
Hey, everybody. Thanks for being here. I want to start out with quickly a disclaimer that I'm going to say some stuff today that's going to sound critical of entities and organizations and people, and it's really not meant to be that way. This is all just observations about the system that we're all operating in here. So please, if anything sounds like an attack on anyone, please note that that is not my intent. So that's it. So we'll just jump right in here. For any of you who are not familiar with the salt marsh harvest mouse, it is a very cool species. It is endemic to the San Francisco Bay, and it's actually the only mammal in the world that is entirely restricted to coastal marshes. So it's a very special and unique species. But of course, our San Francisco Bay estuary is highly degraded. So the species has become state and federally endangered, primarily due to habitat loss. But being a marsh species, you can imagine it is quite well adapted for this habitat type. It can swim very well. It can drink seawater, eat very salty plants. So it's an awesome little guy. And because most of our marshes are dominated by pickleweed here in the San Francisco Bay, they are also very highly associated with pickleweed marsh. And I am sure that none of you here today would ever ask me such a question about this sweet, sweet little angel. But I can't tell you how often I hear this question from folks, and it gets harder and harder to answer without being totally annoyed every time. And you know, the answer that I give them really differs depending on who it is. You know, it may be because it's a really cool species that's unique and it's our, you know, local treasure, or because it's a food item for the species that you actually care about, or because it's part of your permit requirement, and if you don't, your project's going to get shut down. So there's a lot of different reasons why we might want to care about the mouse, and of course, you know, that differs across us. But one of the problems with caring about people, you know, questioning the value of the mouse kind of constantly is that its monitoring for the species has kind of been a little bit devalued. And on top of it being, you know, a little bit devalued, it's also difficult. So the recovery plan for the salt marsh harvest mouse mandates monitoring for the species for recovery purposes, but it's not really delegated out to any entities. On the same vein, there is no entities or individuals that are specifically tasked with providing guidance for a coordinated effort with monitoring. And also, across the years, a lot of different people have done a lot of different work out there, and we've, it's difficult to kind of collaborate and bring together these disparate data sets that were collected under different methods. The recovery plan actually gives pretty good guidance on how often and where trapping should occur. But again, this has not been delegated out to any given group or agency. But there are 3 groups of people that generally do perform monitoring on an annual basis. Our state and federal agencies, CDFW, U.S. Fish and Wildlife Service, do monitoring. They don't do as much as you probably think that they do, and they only work on their own properties. They're really constrained by bureaucratic mandates and funding issues, and they're also constrained by their own mission. Academic researchers have a lot more flexibility in what they want to pursue in terms of research and monitoring, but they have issues with getting access to sites or getting access to permits. And of course, there are folks like me who work for consultants who do some monitoring, but any monitoring must have a research impetus. I can't just go out and trap a site because I feel like it. I may have issues getting access to sites. And of course, we're really constrained by whether or not we have funding. Oftentimes, monitoring only happens if a client is willing to fund it. The centralized guidance. I think people probably assume that the U.S. Fish and Wildlife Service is kind of behind the scenes saying, okay, we're all going to do these sites this year, and this is who's doing what. That is not happening. That is not a criticism of anyone who's working at the service, but they just do not have the time or capacity or staff to take care of that. And at the state level also, that's not really necessarily happening. No one is specifically assigned to it, although some folks are working on trying to do it. And again, even though the recovery plan lists the requirements that we must do this amount of trapping in these areas and have these results to achieve recovery, no one's really, necessarily tracking that or coordinating it or scheduling it. So that's another challenge for monitoring. And then finally, inconsistent methods. I don't got to tell you guys how hard it is to try to reconcile data sets that were collected in different ways. But this is something that we have been working toward in the past decade or so. But what are the consequences of this? What really happens if we don't monitor as much as we want to? Are bad things actually really happening? When we don't have good monitoring data, we really can't track extirpations or understand why they occurred. What you see up here are 2 marsh areas that we surveyed during the range-wide survey last year. If you're not aware, we did a big survey effort last year. I'm not going to go into it, but we surveyed 55 sites with live trapping, 29 sites with fecal DNA, and I'm going to kind of touch on it here, but we're not going to go into it, but you can talk to me about it later. 2 of the sites here, Corte Madera Marsh and Hoffman Marsh, are 2 of the sites that we tracked there during that survey, and they're both areas that are pretty well isolated from any other potential habitat. At Corte Madera Marsh, I caught a single house mouse, and at Hoffman Marsh, I caught a couple house mice and a couple of rats. What you see here are the trap locations. This is data for one day. The blue points are just open traps, yellow is tripped or visited, and pink is a capture. And you can see that at these sites, we really did not capture a lot of stuff. At Corte Madera Marsh, the last time this area was trapped, as far as I can tell, was in 1990. Before that, it was 1971. 1971 was the last time we had a positive hit. So it really seems that mice are gone from Corte Madera, but we don't know when that happened. We don't know why that happened. We have these huge gaps in data that are really precluding us from trying to understand what went on out there that caused them to go away. On the other hand, Hoffman Marsh seems to potentially have been a built marsh. So the mice may have never been there to begin with. So we're trying to figure out why these mice are not at these sites is really difficult when you have a lack of data or a lack of monitoring. In a way, inconsistent monitoring can almost be more misleading than having no data at all. This is a site here. You can see it's not with the light here, it's hard to see, but the pink dots there are all captures. This is a site that we had the most captures at during the range-wide survey. Last year, we had almost 40 animals captured there, which is very high for that number of traps. This year, we went back, we being CDFW, not me, CDFW retrapped it, and did not catch a single salt marsh harvest mouse. This is almost certainly due to our crazy winter that we had, but it really kind of illustrates like how misleading it can be if you're only trapping during certain years and you don't have consistent data. Looking at either of these years separate from each other, you could potentially have a hugely different idea of whether or not salties are present at this site. And teasing out these weather issues has been really difficult, something we've been working on for years, and it's really difficult because, again, our data sets are so disparate and not robust. This is a site that I've been trapping in Marin County for almost 4 years now. The gray bars there you can see are inches of rain between the trapping periods. We started trapping it in 2020. You can see we had very high numbers of mice in the managed ponds. We had pretty high numbers of mice in the tidal ponds, and those have generally been going down over time. So even with like a pretty robust data set, this is twice a year trapping for almost 4 years at this point, and it's still like pretty hard to kind of tease out some of these patterns. And we had this big rain year this year. Looks like numbers are going to go up, but it's going to be really interesting to see how that impacts stuff over time. So I'm really excited to keep trapping this site. We're doing it in October if anyone wants to come out. But illustrating that we have all these conflicting factors that are really impacting salt marsh harvest mouse numbers, aside from habitat. We have all this other stuff that's playing into it. So what does this mean for the rest of the marsh community? We're looking down the barrel of climate change. We know that sea level is going to rise. We know that storms are going to become more intense. And in my opinion, the salt marsh harvest mouse is much more vulnerable to a lot of these potential threats than some of our other species. They are much less mobile, much less able to move out of areas and move back in afterwards. Now, what you can see here on this video, which is kind of hard with how bright it is in here. If you look in the little box there, you'll see a salt marsh harvest mouse climbing up into the Grandelia and during that high tide period, and then coming back down afterwards. We've kind of dealt with this issue of risk to mouse on the marsh plain by kind of assuming that they are moving up into the uplands of T-zone during these extreme events. I did a postdoc study looking specifically at this, trying to figure out how high into the uplands are mice going, so that when we're designing tidal restoration, we can say we need at least 50 meters of upland, because that's how far the mouse goes to get out of the water. And during the study, oh, my pictures got messed up there. During the study, what I found was that the salt marsh harvest mice did not go into these upland areas. They actually stayed down in the tidal marsh areas. So they chose to go into my floating traps rather than go into the uplands. On the other hand, our upland specialist Western harvest mouse was found almost exclusively in the upland areas, so avoiding those tidal areas. And then we have in the middle here our generalist house mouse, which was distributed across the areas. So what this really kind of, for me, it really changed the way I think about the mouse and habitat use. We've kind of really focused on, the mouse is living in the tidal marsh because it really needs pickleweed and it needs these certain characteristics. But everything we know about the mouse, I've been learning over the past decade, is that there's so much more adaptable, flexible than we really thought that they were. And so really it makes no sense that they're not going into the uplands, right? Like they can eat the grass seed, they can eat stuff up there. They're a mouse. They're the most, adaptable kind of animal in the world. So why are they not in these uplands? And, my postdoc data really kind of indicated that it's not anything about the habitat that is specifically, shaping this habitat use. It's the competition that's in the uplands. So we have these other more aggressive species that are more able to exploit and protect those upland areas. And salties are not going up there because they're going to get beat up if they go hang out. So, for me, that really kind of changed how I think about things, of focusing less on this, T-zone upland area, and really thinking more about making refuge for them out on the marsh plain. And, even if my whole postdoc was totally messed up and my results are totally wrong, which is, totally possible, it's probably not, but it could be possible. Even if my study was totally wrong, some of these animals live hundreds of meters from an upland edge, and expecting that they're going to have to run every single time a big tide comes to get up to the upland edge to find refuge is really just not a sustainable practice for the species. This is a video here at Bothene Marsh during a king tide event, showing just how intense some of the flooding can be in some of these areas. And I would argue that, salt marsh harvest mice are especially vulnerable to some of these issues, especially this flooding. And if we're seeing those guys kind of pushed out of an area, we know that something has gone pretty wrong, because that means there's no refuge for them whatsoever, that they can, persist in these areas. And that doesn't bode well for any other species. If our most vulnerable species is starting to suffer, that means it's only a matter of time before the next species start to have issues. This is Alameda Creek, or Alameda Flood Control Channel. The larger picture you see is the flood control channel on the left, and a salt pond and the Coyote Hills on the right. This was, we trapped this for the range-wide survey. There were salt marsh harvest mice here during the big storms this winter. This area was completely scoured out by floodwaters. The picture you see on the left here are huge trees that were tossed up onto the marsh. Imagine being a 10-gram animal and trying to cope with that. Like, you can't deal with that. And so if any of our salties survived here, it's only because they either moved over into the salt pond or found some other place out of that tidal area to get out of the water. So these extreme events that are happening, that we've been anticipating, are already happening. We're already seeing the consequences of that. And the consequences are that we're seeing animals drowning and dying. This is a pocket gopher, not a salt marsh harvest mouse, but this guy drowned during this thing, and his little body washed up here onto the road. So we're already seeing the consequences of, this lack of refuge for salt marsh harvest mouse in our area. These are the photos from, well, the video's not going. Oh no, how sad. These are the photos from the Twitter post that Julie referenced on the first day. This top photo here on the left is a dead house mouse. The bottom photo there is a dead vole. What you would see here if this video was working is me panning over. This is Santa Venetia Marsh, which is a pretty resilient marsh. It's got good veg. There's an area behind a little levee that is a great little muted tidal marsh. This video would pan around. You would see there's a lot of this stuff flooded, and it kind of pans down into some veg where a salt marsh harvest mouse is hanging out. He's fine. He's alive, not dead. Hanging out and hiding in the vegetation for the waters to recede. So these guys are adapted to coping with this, better than some of their upland associated counterparts. So that is a good thing, as long as we have that refuge for them. Even the young actually are quite adaptive. So this is zooming in here on a little nest that got flooded. I was out here, this is at Sonoma Creek. I went out to photograph the king tide, and I heard some little beep, 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 beeps. And I found these two little salt marsh harvest mice who don't even have their eyes open, who had climbed up the pickleweed and out of their flooded nest to hide, or to get out of the water, to protect themselves during this king tide period. So even at a very young age, they are adapted to coping with these issues. I don't think I've ever showed these. This is from 2020, and I don't think I've ever showed this at a presentation, so you guys are getting it first time here. Here, I can play it again. But even the little guys, like, they know what to do when the water comes, as long as they have the, habitat there to cope with it. So that is a little bit of a silver lining. And along with that, another silver lining we've had in the past couple of years is that we have found that they're able to use much more narrow marsh than we previously anticipated. The recovery plan says 100 to 300 meters is necessary. During one of the island pond restorations, we found mice nesting in habitat as little as 10 meters wide. So this bodes really well for some of these, very narrow strip marshes in the South Bay. It bodes really well for these upland marsh migration edges. Even if we're only getting a small edge of migration, marsh migrating, the mice may be able to use that for nesting habitat, or at least as dispersal habitat to move between larger patches. But we really need to have more robust monitoring to deal with some of this stuff. So what I'm showing here is just the North Bay results from the range-wide survey. The circles are live trapping, the triangles are poop DNA, red is no captures or detections. It's really hard to see here with the lights, but and blue is detections. And so as we move into an era where we're seeing our marshes migrate, we run into the issue of this upland habitat, which is, potential future habitat for salt marsh harvest mouse, which is not protected because it's not technically their habitat yet. And the ESA does not protect potential future habitat. But we know that's really important for the mouse. Even if they're not using it right now, it's important for them for their future. And so, it's gonna be really important to monitor these areas for use, because if the mice start using them, then maybe we have an excuse to protect it, even though it's not, a wetland of the United States. So I would really encourage folks to be, vigilant of those areas and monitor them. Even if you don't necessarily think that they're there yet. One of the lessons we just recently learned, got results back from, we put poop stations out at Cullinan Ranch, which is, you guys are mostly probably aware of it, a restoration area. This is the Cullinan Ranch East, where they built those cool islands. And you can see the islands and you can see this pickleweed at the bottom of them, but they haven't breached it yet. Most of the area is unvegetated. I wanted to put traps out there to basically document absence. We put the traps out there, and lo and behold, we find there are already salt marsh harvest mice utilizing the pickleweed down there at the base of those islands, which the refuge was quite surprised about. We're now going to try to do some additional live trapping out there to look at these islands, figure out which of the islands are they using. But if we only trap once we think it's good enough, we can't track when animals are coming in. We can't track what features of a restoration or of a migrated marsh area are facilitating the salt marsh harvest mice colonizing or moving in. So it's really exciting to take a look at this. We're going to try to trap it in October. Hopefully we'll see if we can make it happen. But you may just remembered that I, just harped on how difficult monitoring is for mouse. And now I'm telling you to do more of it. And it is difficult. You need to have permitted experts, you need to have money on top of all the other challenges that we're already facing. But things are getting easier. The Salt Marsh Harvest Mouse Working Group has spent a lot of time and effort trying to make monitoring more effective here. We have been working on standardizing methods and creating a protocol. You may be surprised to hear that unlike ridge-raised rail, which has an accepted standard protocol, there is no standard protocol for salt marsh harvest mouse. Anyway, when you're trapping, you can pretty much do whatever you want as long as it's approved in your permits, which is the same reason that you can't do a survey to prove absence of salt marsh harvest mouse. There is no such thing as a protocol level survey for mouse. But we are working on trying to standardize our methods and come up with a protocol. We've also been using genetic research to refine some of these lists. We have these long lists of all these characteristics. Say it's a salty or it's a Western, and Dr. Mark Statham at UC Davis has been doing some great work with some genetic testing to kind of weed out some of those characteristics that are not so diagnostic, so we can spend more time focusing on the ones that are diagnostic. They've also been looking at some of the historical records, museum specimens. We've also been trying to utilize technology to kind of make our survey efforts and data management more streamlined. So ArcGIS Online and field maps. You can see here, this is a picture of someone taking a photo of a mouse on an iPad, trying to make things work a little bit better. We'll make doing surveys, more efficient and easier. There's also options for non-invasive surveys. Acoustic monitoring is something that I've been toying around with for about 10 years. It's really difficult. I have not gotten it at all anywhere close to being effective, but it's still an option. Acoustic stuff is getting more efficient every year. We can all now identify birds on our phones. So, maybe it'll get there at some point, but it's an option. Camera traps are cool for collecting observations of life history and activity. If you have someone who's really experienced to review it, you might be able to get presence absence data, but it really doesn't give us kind of any idea of population sizes. But again, this kind of technology stuff is advancing at rapid speed. So who knows what we'll have with it in a couple of years. But one of the most promising new methods that we have was developed by Dr. Mark Statham and Dr. Cody Aylward at UC Davis as part of Dr. Aylward's PhD research, which is fecal DNA testing. We tried out eDNA. We've had some mixed results. We haven't spent a lot of time kind of going back to it, but the fecal DNA has been super effective. So we basically put out these little boxes that have food in them. The mouse goes in, the mouse poops. We take the poops, we genetically identify them, and then we can say salties are present or not. And if you put out an array of these, you can kind of get an idea of relative abundance too. If you put out 10 traps here and 10 traps here, and this one has poop in every trap, and this one has poop in one trap, you can say, well, there's probably more salties over here than there over here. And this is pretty affordable. It's much cheaper than a full live trapping survey. They were still required to do permitting for this to get it to do recovery permit. I don't know how that might be in the future, but it's something that could potentially really open the door for a lot more comprehensive and higher coverage of monitoring throughout the range of species. So that was a lot. I just threw at you a lot of different slides, different stuff. So I want to wrap it up here. Salt marsh harvest mice are not being monitored as closely as a lot of people probably think they are. At one of the first meetings that I went to for the WRMP, someone said to us, well, we don't need to put mouse in there because the refuge, the U.S. Fish and Wildlife Service is already monitoring them. And that's just not always true. Like sometimes they have the time, sometimes they have the money, but sometimes they're specifically told, like, you're not doing mouse monitoring right now. And so we cannot rely on the folks that we assume are doing monitoring to provide this monitoring for us of really important data that we all need. And again, the systematic monitoring is challenging. So the more that all of us are participating and planning and integrating this into the work that we're doing, it's going to create a better data set for us. As I showed you with Corn Madera Marsh, these gaps in monitoring really reduce our ability to assess the threats and the changes to habitat that are occurring and how those habitat changes are impacting the mouse. Because again, these guys are less mobile than other species. They are really sensitive to threats more so than other species. They cannot disperse through cities. They can't disperse through water. If they're gone from a site, they are gone and they're not coming back unless we put them there if the site is isolated. But again, they can use narrow areas, including these narrow strip marshes, potentially these migration upland edges. But again, they're still not going to be able to persist if there's not some kind of refuge for them. And again, this lack of monitoring reduces our ability to gauge the progress on restoration. So looking forward, as we're planning restorations, not only worrying about what's going on in the areas that exist that we don't know much about, we need to worry about our restorations too. And we should be planning for at least some kind of monitoring for mouse as we're doing these, and anticipating, trying to understand when they're coming in, not just waiting 5 or 10 years and then coming and seeing if they're there. Because we want to know what is it that's bringing them there. We need more comprehensive monitoring. So we have great tools. It's got cut off, which is the crux of it, cut off from the bottom. But hopefully I have convinced you that the salt marsh harvest mouse is actually one of the best indicator species that we can have out here for monitoring in the Bay. These are the guys that if they are lost from a site, something has gone very wrong, and it's going to be bad for the other species soon. If you survey for rails, you leave, they're gone, they get wiped out, and you come back in a year and another rail came back, you don't even know that they were gone that whole time. If you survey for a mouse, they're there, you come back next year, they're gone, you come back the next year, they're gone. Next year they're gone, like they're gone, so it's a really good indicator that either something catastrophic happened that needs to be dealt with or accounted for, or that the habitat has changed so much it's no longer suitable for mice. And that's data we really need to understand as our habitat starts to change much more rapidly. So monitor more for mice. I would like to thank all the collaborators and funders who have really supported all of the work that we've been doing over the past 10 years, especially with the range-wide survey. We had a huge amount of effort helping out. I also specifically want to call out Lorraine Barthman-Thompson, Sarah Estrella, Melissa Riley at CDFW who have done a huge amount of work to keep the mouse work group going and push for a lot more monitoring and standardization, even though it's not necessarily part of their job. They go above and beyond to make this happen. So big shout out to them. If you want to chat more about mice, please feel free to hit me up anytime. If you want me to just ID a mouse for you, just send me a picture of it. You can send me pictures of mice anytime. It's great. I love it. And if that, I'll take any questions if I have time, which I maybe don't. Later. Yeah, later.
Awesome. Thank you, Katie. I love those visuals. I can't wait until we hear about how many mouse poops you've counted. Okay, next I'd like to introduce Caitlin Crane. I work with Caitlin all the time. She's probably sick of me, but Caitlin is a lead tidal wetland ecologist in the Resilient Landscape Program at the San Francisco Estuary Institute. She also happens to be the lead scientist for the San Francisco Estuary Wetland Regional Monitoring Program. Gosh, after Caitlin's talk, if only there was a coordinated estuary-wide funded regional monitoring program for the tidal wetlands.
Imagine.
That's
Imagine.
crazy.
Great segue. Well,
Ooh.
thank you. So yes, I was thinking the same thing, that there's just a lot of synergies that are emerging here through the talks that I've heard over the last couple of days. And I'm here to talk about the Wetland Regional Monitoring Program, the WRMP. And the timing of this this workshop is excellent for getting this young program off the ground, which is what we are currently in the stages of doing, while keeping our eyes on the prize. And that is thinking forward about the managing this system for adaptation and migration and understanding migration, planning for migration. That's one of our key management and adaptation tools that we have going forward. So the idea with monitoring is that you establish a baseline, collect data using standardized protocols, which we're hearing a lot about the need for, collect similar data over time to track change. And in the case of the WRMP, we're very much designed around the idea of we want to collect data that we can translate into information that matters, that people can use to adaptively manage the system. And so it's no surprise that this type of program has emerged in this region with all these forward thinkers and folks like you. I mean, in fact, many of the people behind the workshop today have been on the front lines of bringing this program to fruition, including Christina Toms, who's our chair of our technical advisory committee, and SFEP, where we are today. So it's no surprise that this program has emerged to answer the questions that this workshop is identifying that we need. So kind of an outline of what I want to go over today is a real brief overview of the background and progress of the WRMP. We have been in the design, planning, and dreaming phases for a long time. You've probably heard of the WRMP in a variety of ways over the years. And that's because the program's been funded by the EPA through Wetland Program Development Grants to SFEP to get this program designed and going. But we're now about a year into a funding from the San Francisco Bay Restoration Authority to move from this planning phase into implementation. And so I think that one of the goals of mine today is to sort of brief you on where we are in actually getting the program off the ground. And what are our near-term science priorities and some of our early achievements. And then to get into what kind of monitoring is underway and could happen in the future with program funding, continuous program funding, that's really relevant to marsh migration. So the WRMP, the goal of the WRMP is to deliver coordinated regional information, I'm covered up, science to inform stewardship, restoration, and adaptive management of the San Francisco Estuary wetlands. And there it is right here in our mission statement, adaptively manage. And so we're doing that through establishing a monitoring site network. And that's an early achievement that I'll tell you a little bit more about later. An open data sharing platform so that we can take the standardized data that's being collected around the region and share it and translate that into usable information. And we are backed by this comprehensive science framework that's gonna help us decide what to monitor when. And I highlighted here at the bottom, a major goal is this inclusive, collaborative, and transparent approach. And that's something that I've been most impressed by coming into this program. Like, what an amazing group of people have been hard at work, working together to bring this program to fruition. And you know, I think of this really, I'm reporting on the WRMP today, but this is really our regional monitoring program. So the things that we have going into the plan and into the program come from you and your needs. And so again, this workshop's been a really great iterative process for us understanding what kind of information is going to be most useful going forward. So as part of this collaborative process, we are guided by a steering committee that has representation from many different agencies and a technical advisory committee that's helping develop the real science backbone of the program. The program is implemented through San Francisco Estuary Institute, where I am, and the San Francisco Estuary Partnership. So we have a lot of players trying to pull this program together. And again, we're young, we're getting this off the ground, and it's a work in progress. We're focused, and I'm going to tell you today again about the monitoring network, which we've just recently gotten designed and off the ground, and the science and data sharing platforms. So this is trying to summarize a lot of planning into one slide, but this is to illustrate the process by which we've come to where we are today. So the rainbow of approaches takes us from the guiding and management questions that the program plan published in 2020. The steering committee helped to deliver, these are the guiding questions, things like where are the tidal wetlands and how are they changing? How are external factors impacting wetlands? How are plants and animals being affected by change? What new information do we need? And how are the health, and again, I'm covered up here, you can read it better. Health of the wetlands impacting people. And so we've gone through this process from the guiding and management questions. So what do you need to monitor to answer those questions? So we have our monitoring questions that point us to indicators or metrics that we're actually going to be monitoring over time. And we're in the process right now of establishing these standardized protocols. And this work happens through work groups, again, pulling in this knowledge from the region and from a range of different experts. We've established a geospatial SOP for mapping habitats, and we have a fish and fish habitats SOP that's been completed. We're currently near to completion on a vegetation SOP and a hydrogeomorphic SOP. And the people and wetlands work group will be developing standard operating procedures as well. So these are large products to be coming out of the WRMP in this calendar year. Because this program is modeled on an EPA RAMP framework, we are focused on monitoring at different levels. So level 1 being the region-wide remote sensing mapping. This is really appropriate when you're trying to answer questions on this large regional scale. Level 2 is a rapid assessment. And in this case, we're focused on the California rapid assessment method. And level 3 is what you think of, at the site, the data sons and loggers, the individuals going out and monitoring more fine scale change. And that's really appropriate and necessary in some situations to look for mechanisms and details, but it's integrating through these different levels that really enables the greatest amount of information gathering at this large regional scale. So here is our WRMP priority monitoring site network as identified in a memo that was approved through our steering committee earlier this year. We are distributing our sites of interest across the estuary and the different sub-embayments that are colored in various colors. The hatch lines identify the actual network. So we've got Sassoon, Napa Sonoma, the Novato Galinas, Wildcats, Alameda, and Santa Clara Valley. So within each of those subregions or small operational landscape units, we've identified 3 different marsh types that vary in their history and human intervention, degree of human intervention. So this is an example of the Novato Galinas network, where you can see China Camp would be our benchmark site. So this is a historic wetland with relatively little disturbance. Outer McInnes is a reference wetland. So it's a younger wetland. The reference sites are either old restoration projects, or in this case, a fringing marsh that's developed in the last century. And Hamilton Wetlands is an example project site. So we have this group of different types of wetlands all united in this subregion and then spread throughout the whole estuary. And the idea is that we can be monitoring both the ambient background conditions and how they change due to climate change, as well as the changes due at restoration project sites, and put those in the context of this regional change that we're all experiencing and interested in. So there is a lot of questions and approaches to monitoring that we could do at this regional scale. And we're an ambitious program, and we've got a lot of people with great ideas. So we're at the phase of needing to focus. And so we've identified, basically through the sort of distilling the guiding and management questions that we've been given, distilling those down to, like, what do we really want to know first? What do we need to get in the ground, start this benchmark monitoring so we can then track change going forward. So we've identified our near-term program focus to answer the questions: where are our tidal wetlands? How are they doing? And are they drowning or keeping pace with sea level rise? And so that helps us then translate into our near-term monitoring priorities. So we've identified for the program 3 near-term priorities, and that is our level 1 region-wide mapping of the tidal baylands. So this is affectionately known as the Baylands Change Base Map, mapping the habitats of the baylands so that we can then look for change, look for migration, look for shifting distribution patterns. Secondly, to answer how are these marshes doing, we're focused on using the California Rapid Assessment Method, CRAM, to look at marsh condition. And third, to address whether marshes are keeping pace with sea level rise, we're going to leverage work from the US Geological Survey to monitor a regional network of SCT, sediment elevation table marker horizons, that look at really fine scale change in accretion and elevation at a specific site. So these 3 priorities help answer our driving questions, and they also map onto that level 1, 2, 3. So I'm going to tell you just a little more about these near-term monitoring priorities and one of our early achievements, which is the nearing completion of the Baylands Habitat Map, or the Baylands Change Base Map. So this effort was funded by the EPA. It's been undergoing for several years, guided by the geospatial SOP, where we're using remote sense data and machine learning using e-cognition software to have the program actually map and identify the different habitat types within the baylands. Of course, using expert input to clarify and make the habitat types more accurate. But this approach not only establishes a new base map of the baylands at this regional scale. This hasn't been done since 2009, but it establishes these standard protocols that will continue to happen every 4 to 6 years, so that we will be able to track change in habitats, not due to a difference in imagery or different people analyzing the imagery, et cetera, but actually tracking change. And that's really what the program is designed to do. And it's exciting that we're about to just be able to share the results of this new base map at this regional scale. The base map then allows us to ask all sorts of interesting questions about wetland characteristics across the landscape. And so some of the examples, analyses that we can do from this base map include things like understanding marsh gain or loss. So where restoration projects have actually revegetated and created new wetlands, not just where they are planned or have been breached, but actually new wetland occurring. We can think about patch size and configuration across the landscape, percent vegetated marsh, elevation capital, complete marsh, and that's something really relevant to today's discussion. So, if you were here yesterday, you saw Ellen Plain, a colleague at San Francisco Estuary Institute, who is working with the Shoreline Resilience Framework, which is funded by the Army Corps, and is utilizing the WRMP's base map to start to do some of these metrics and analyses, including things like complete marsh. So it's really exciting that this, the WRMP's monitoring data can then be used by a number of partner organizations. For, in terms of marsh condition, we're using CRAM. This is a protocol that's been really well developed and vetted through the region and is being used elsewhere in the state. So we don't want to recreate the wheel. Let's use a protocol that's in place and it has ways that we can interpret the data. So this figure here looking at good, fair, and poor marshes is an illustration of how a CRAM score, a rapid assessment, can be interpreted within a regional distribution of other marshes. And the CRAM score here you can see is created by summarizing 4 different attribute types. And I just wanted to show that because one of the attribute types is the landscape and buffer. And so buffer extent and quality is part of the sort of qualitative assessment that goes into CRAM and is really relevant for understanding, marsh migration space. So finally, our third near-term priority is thinking about fine scale accretion and elevation change using these SETs. And again, this USGS already has and NOAA and others in the region have SETs throughout the region that we can kind of leverage for help to support new SET that going into new locations where there's data gaps. But the theme of the WRMP is leveraging what's already been invested in the region and using that incredible base to create more information. So future potential monitoring that's really relevant to marsh migration space, and there's a lot of possibilities depending on how the program grows, but we've already developed this geospatial SOP that highlights transition zones and marsh migration space mapping. We're not doing that currently because the region has some excellent maps already created, but we have recommended protocols and could serve as a hub for continuing this mapping going forward. The vegetation SOP recommends a remote sensed region-wide mapping of vegetation alliances. Those are the dominant plant types. And because wetlands are biogenic habitats, they're created by the plants themselves, understanding the shifting distribution and dynamics of the plants really feeds up into understanding the habitat types and what's going on with the wetlands. The vegetation work group also recommended a special study that I'll just show you a quick picture of that's really specifically focused on tracking change in the transition zones. And that's because transition zones, as we've heard, is a place of greatest plant diversity for within the wetland and is the place we expect some of the greatest change due to shifting physical drivers. And by focusing on the historic benchmark sites and looking at transition zone change, we may actually get to the mechanisms behind how these wetlands are going to move. Is this a gradual linear process? Is this a stepwise function where plants are moving more dramatically when there's an extreme event, et cetera? Also, the hydrogeomorphic SOP in particular, I'd say monitoring elevation, regional DEMs, which are the sort of region-wide maps of elevation or site-based elevation maps, water surface elevation. These are the drivers of wetland migration. And so having consistent data tracked over time could provide some leading indicators of where we expect change to occur. I feel like we are really getting tight on time. So I'm just gonna point out again this special study that highlights how we can specifically be monitoring the vegetation across transition zones and wetlands, but not go into the details now. So what's next for the WRMP? We're going to be finalizing these standard operating procedures. We're working hard to get our initial monitoring plan for the region off the ground or finished so that we can transition into an implementation plan. And in the meantime, we will continue to serve as a regional hub for monitoring and bringing people together to discuss the opportunities and the best approaches, and continue to be involved in these interesting conversations like we're having today. So thank you very much.